саркоидоз и туберкулез, достижения иммуностимулирующей терапии имеет профессор Тревор Маршалл. Professor Marshall, you are welcome. For first presentation, you have 20 minutes.
Thank you, Leonid. Okay, this is going to be interesting. Well, I'm going to talk about sarcoidosis and Mycobacterium tuberculosis, and what we have discovered from a number of years now, nearly a decade, of use of an immunostimulatory therapy in sarcoidosis. I'm going to start with a quotation from Professors Yehuda Schoenfeld and Noel Rose, both very eminent autoimmunologists, who in 2004 made the observation in a book called Infection and Autoimmunity that everything is infectious until proven otherwise. A pretty bold statement. Now I'm going to extend that in 2012 to say that we can say that the human microbiome causes human chronic disease and predisposes for much infectious disease. That's what I'm going to be talking about today. And I'll offer no less than the eminent medical journal, The Economist, as part of the hypothesis here. And The Economist carried in August a very, very good article, Microbes Maketh Man, talking specifically about the microbiome and the various diseases which have already been linked causally to the microbiome. And I want to point out to you what a wonderful illustration has been used here by the artist that drew the front cover image. You'll see that a few times in my presentation. But if you want something a little bit more austere, we'll talk about the National Institutes of Health NIH Human Microbiome Project, which ran from 2007 to 2011. It's finished now, and what they found was that man is a superorganism. The HMP identified millions of genes belonging to thousands of species of microbes which live in and on a healthy human body. So for the 25,000 human genes that fundamentally control our existence, there are greater than 1 million bacterial genes, plus a whole stack of viral genes, and also the fungal genes, and of course the phage genes. And it is our microbes that make us genetically unique. There is more difference between the microbiome of everybody sitting in this audience than there is between the genome of everybody sitting in this audience. Our microbes make us genetically unique. Metagenomics is the study of those microbes. So where do you get the microbes from? Well, firstly, you get them from your mother, and at birth babies are born dirty with a gut full of bacteria. They very quickly pick them up. Within the first couple of weeks of life, they've picked up most of the microbes of the extended family. Some very good work has been done by Dave Rollman's group at Stanford on that era of life. Then you really can't avoid them from that point onwards. If you look at air, which we all breathe, we can't avoid it, and you sequence the organisms, the DNA, which you find in air, well, in a hospital, you would expect maybe in a hospital that you would find a large percentage of bacteria, about 86%, in addition to human DNA, fungal, animal DNA, plants, and viral DNA. But a huge contribution from bacteria, much greater than the human DNA that's floating around in a hospital. But it's surprising that you find a very similar thing in the house. Inside a standard house in San Diego, the Craig Venter from the Craig Venter Institute found that once again the air was containing about 96% of the DNA in the air, 85% of the DNA in the air was due to bacterial DNA. You also pick up bacteria from travel, and particularly from international transport of foods. Over the last 5 decades, our food has become an internationalized commodity, and with it, it brings the microbiome of the region from where the food comes. So a decade ago, I was looking at sarcoidosis and looking at sarcoid granuloma, and something hit me about this sarcoid granuloma. I noticed that most of the lymphocytes, which are the black stained cells, of course, most of the lymphocytes were around the periphery of the granuloma, as if they were expelled to the periphery and not a key part of the process that formed the granuloma. About 90% of the cells in the body of the granuloma are monocytes and macrophages, but particularly monocytes. And you know, that seemed to point to me that innate immune activity, the protection within the cells was likely to be key in granuloma formation rather than adaptive immunity, the T cells and the B cells, as everybody was suspecting at that time. Well, there was a very elegant study done by Wirostko, Emil Wirostko's group at Columbia University in the 1980s, and they used electron microscopy to look at the cytoplasms of cells, of the cells from the immune system. This is a monocyte, and it's taken from the eye of a sarcoidosis patient. And you can see hundreds of tiny coccoids here which stained. They used a urinal acetate stain for nuclear material and found that there were many colonies of microbes in the cytoplasm of these cells of the immune system. The very cells, the monocytes that are supposed to kill pathogens, were being parasitized and providing a safe harbor for pathogens. The same thing happens for lymphocytes. This is from a juvenile rheumatoid arthritis patient with a large cluster there, looking something like a I'm sorry, my mind just blanked out then. But there's a large cluster and also several linear clusters or linear DNA segments as well. It's very difficult projecting from the side of an image, I'm afraid. Sorry about that. Let me apologise in advance. So what happens when pathogens manage to get inside a cell? Well, that has been studied. And in China in 2003, a DNA microarray was used to study an infection of a macrophage by Microbacterium tuberculosis. And what was found was tremendous change in the operation of the cell. There were 25 genes upregulated, 341 genes downregulated, and the VDR nuclear receptor, which is the one that we have found is the focus of disease activity in sarcoidosis, was downregulated 3.3-fold by the infection of Mycobacterium tuberculosis. In other words, once a pathogen gets inside the cells of the immune system, it changes the way that the immune system works. The microbiome messes with our own human biology. Inside the cell, many metabolites are affected by genes from the microbial metagenome. And the sum total of all these interactions gives rise to the symptoms which are suffered during chronic disease. The sum total, we're not talking about one or two symptoms, we're talking about thousands of symptoms. The genes from microbe A and the genes from microbe B might lead to a particular dysfunction, which I'll call X, whereas genes from a number of other microbes combine their dysfunctions in order to create the symptom, which I'll call Y. The genomes in the microbiome accumulate gradually during life. We're born with some, but we pick them up gradually based on our environment. Genes from the accumulated metagenome determine the clinical dysfunction and therefore disease symptoms. And the interactome is huge. The interactome is the name given for all of the interactions within a cell. And when we look at those which are involved in disease, it's absolutely huge. This is a study which was just performed on CSF from chronic fatigue syndrome patients up here, chronic Lyme disease, PTLDS patients here, or PTLDS patients there, and healthy controls up there. And out of about 2,783 total proteins measured in CFS, a similar number for healthy controls and PTLDS, these were absolutely unique to each of the disease processes. For healthy human beings, there were 724 proteins that weren't found in either CFS or PTLDS. In PTLDS, there were 692 found that were unique, and in CFS found there were 738 that were unique. We're not talking about a single pathway here. We're not talking about something simple like JAK-STAT that we can knock out. We're talking about an incredibly complex set of interactions which lead to these incredibly complex diseases we call chronic disease. And of course, genetic science is still struggling with that complexity. This is a paper published just last week trying to differentiate healthy controls from sarcoidosis patients with about an 80% accuracy, which they used 20 genes, which clearly is not nearly enough. We're talking about hundreds and thousands of total genes. So back in 2004, Professor Alexandra Wiesel translated one of our papers for the Kazan Medical Journal, and as you can see, it says that antibiotics are effective at causing remission in sarcoidosis. Well, that's not exactly true. We were wrong. The patients were in fact being given antibiotics, but they were also being given a drug for palliation, a drug called Olmesartan for palliation. And in the years which have followed 2004, in the last decade almost, we found that the thing that did all the work, that the key thing to knocking down the microbiome, the key element, is knocking down or strengthening innate immunity so that innate immunity, the individual, can attack the microbiome. The antibiotics are relatively ineffective, and we dropped them about 4 years ago. The other thing that's very interesting is that this is unique. The way that the innate immune system works is unique to Homo sapiens, because only in Homo sapiens there's one nuclear receptor which expresses genes for TLR2, which is a key defense for the cytoplasmic region of a cell, as well as cathelicidin and beta defense in antimicrobials, all of which are associated and essential to the intracellular innate immune defenses. In order to survive within human cells, microbes clearly have to knock out the VDR. Microbes cannot exist in the presence of cathelicidins. They are very, very effective antimicrobials. The beta defensins are also effective, and TLR2 is a key signaling molecule, a key signaling receptor. So in order to survive, they have to knock out the VDR. That understanding is key to the next step. And the next step is to find a drug which actually activates the VDR. And what we found was that the drug that we were using back when we published the article from Kazan, we were using this drug as a palliative because it's designed to knock out the angiotensin II type 1 receptor, the angiotensin receptor. It's a sartan, and it was designed to reduce blood pressure. But if you change the dosing a little, if you change the dosing so that instead of giving one dose a day, shown by the green pulse here on the graph, we give multiple pulses at 4 to 6 hour, multiple doses at 4 to 6 hour intervals, you can see we build up a basal level of concentration in the bloodstream, and that changes the operation of the drug. At that point, it starts to activate the VDR. And that's important because in normal healthy individual, the endogenous steroid calcitriol will activate the VDR, and that will then cause gene transcription of at least 1,000 genes that the VDR is totally responsible for. But when the microbes come along, they produce toxins, and I mentioned gliotoxin, we'll see that on a later slide. Gliotoxin is one of the identified toxins that knocks out the VDR, stops it from being activated, stops the endogenous steroid from activating it. And once we applied that to a human cohort, we got some amazing results. And this is one of my colleagues from the Autoimmunity Congress in Asia last year, and the key that was really interesting was the coupling of cognitive and memory difficulties with the primary condition, the sarcoidosis. As the sarcoidosis disappeared, so did cognitive and memory difficulties. Now I have three quick case histories here. All three of them were pretty sick individuals. This is a male born in 1939 with rickets. He's now 72 years old. Had two various problems, and then ended up with two bypasses, two quadruple bypass surgeries, plus a cardioversion, and also had a pacemaker implanted, and then was really sick in about 2004, had 2,500 ml of pleural infusion. But after began Olmesartan, you'll see that there are very, very few very few entries. The patient's doing very well, no oxygen anymore, and no sarcoidosis remains clinically at this point, as the patient's enjoying life. This is a female, AC, who as a child had various problems, tonsillectomy, kidney inflammation, breast lumpectomy postpartum, goiter, enlarged spleen, and once she began Olmesartan in about 2005, over the 6 years intervening, has basically cleared and is now active in community affairs, enjoying life again at 61 years old. This is a case history of somebody who actually lives in Alaska, was on oxygen, very seriously on oxygen, was prescribed end-of-life care in 2006. Whoops, wrong button. I meant to press the laser pointer. There you go. Was prescribed end-of-life care, went instead to the experimental therapy on Olmesartan, and now doesn't need oxygen, is very active, just did a sea kayaking trip without any oxygen at all, and walked a half marathon without any oxygen at all. So that's the sort of improvement that we're seeing when the innate immune system is reactivated to help people recover. The problem, of course, is immunopathology, because when the host resistance kicks in here, then there is also some damage caused by the immune response, the immunopathology, and that has to be managed. And immunopathology changes with time. Unfortunately, the symptoms get worse before they get better. So you can see here there's a peak in the ANA of this arthritis patient, and this ankylosing spondylitis patient also gets worse before they start to resolve. So let's go back to the start again. Everything is infectious until proven otherwise. So the question is, why do we treat infection with immunosuppression? Palliation of symptoms usually makes an underlying infectious condition worse over time. We now can routinely find microbial DNA in healthy human blood using modern sequencing techniques. So surely we have to adapt the clinical paradigm. We have to understand we're dealing with microbes inside people and not just symptoms that have to be palliated. Ultimately, this may come down to a choice for mankind between the current paradigm of feeling good, palliation of symptoms, and a choice for health, the absence of disease long term. I'm going to talk more about TB in my next presentation, which is on Saturday midday. So those of you that can get to that presentation, I just want to let you know that I'm going to talk about more about TB in that particular one, and particularly drug resistance and how it relates to the microbiome. Thank you.
Thank you, dear doctor. You have economized 1 minute for two quick questions. Кому угодно задать вопросы, пожалуйста.
Извините, я не понял, каким препаратом лечение проводится?
Where's my mic? We used a drug which was approved by the FDA in America for high blood pressure, called Sartan, S-A-R-T-A-N, a drug called Olmesartan midoxamil. And we found that Olmesartan midoxamil was unique in that when we changed the dosing, it targeted other receptors besides the angiotensin II receptor that it was designed to target. So it was Olmesartan midoxamil is what we found was useful.
Использовался антагонист ангиотензинового рецептора Olmesartan, повышенный.
Дозы препарата и как долго можно принимать препарат? Всю жизнь?
The duration of treatment is very long. Hardly anybody responds within the first 2 years. They start to respond clinically, typically at beyond the 2-year point, and become the disease resolution typically takes 3 to 6 years. The patients often get better beyond that, particularly in terms of cognitive and memory issues. So effectively we're talking about a disease that takes a lifetime to gradually come on, and which takes about a decade to fully get rid of. It's not a quick fix at all. The dosing is 40 milligrams every 4 to 6 hours of Olmesartan midoxamil is what we use. So that keeps the blood levels, if you remember the slide, you can see the blood levels stay below a normal level that is known to be safe with that drug.